Well, good morning, or if you're listening to this in the evening, good evening. We're going to talk about chapter 22 today. This lecture will cover that portion of your material, talking about prokaryotes, the bacteria and the archaea. Remember, we talked about it in lab the other day. The prokaryotes are organisms that don't have a nucleus, right? That's what unifies them. And for many years, we used to lump them into one big kingdom that we called Monera. You don't see that kingdom name anymore because that was an artificial group. That goes back to our cladistics discussion. Putting the bacteria and the other prokaryotes, the archaean, together into one big group and calling them Kingdom Monera was really similar to putting us and the fungus together in one big group and calling us, animal fungus things, some name that we would have got for that. In other words, if you're thinking about the cladogram, the family tree of the Monerans was artificial. It wasn't monophyletic, was it? Right? Because it was actually two different very big branches of organisms that were in there. So the Kingdom Monera, which was lumping them together based on the way they look, like we've talked about, that can be problematic, right? The way things look because they're small. A lot of times the organisms work a clump that way back in the early days. For example, next chapter, you'll see where the protozoans are really in flux as far as what they are. But nonetheless, one of the things I want to talk to you about in terms of the, and of course, remember from Bio 1 that the prokaryotes are broken in now into two domains, right? It's the domain bacteria and the domain archaea. And recall your taxonomy, a domain is above a kingdom. So that means that, and there's only three, right? There's bacteria, there's archaea, the two domains and then ours, right? Eukarya, the other one, the ones with the nucleus. So not having a nucleus doesn't mean you're closely related. It's all relative, right? Because we've now seen that we can break up those things without nucleus into two big domains. So the Earth has three domains, one that we're in, and then the other two that belong to these little things that are everywhere. This is one of those electron microscope pictures of one of those everyday ordinary objects, that they blow up really big and they try to get you to guess what it is. If you look at this and you think about being magnified, I don't remember how many times this was, 20,000 or some incredible number. It looks like that purple thing is coming to a point. And even though it's flat at the top, if you magnified it 20,000 times, if we looked at it with our eyes, that would be very, very sharp. This is the tip of a needle. And what those little yellow things growing all over, or found all over the tip of that needle, are prokaryotes. Some are maybe in the domain bacteria, some are in archaea. Now, being old school the way I am, I sometimes slip back and forth between calling all these prokaryotes bacteria, so I'll try to catch myself, but if that becomes confusing, then of course let me know. I'm a victim of history, right? What's really cool about these organisms, we were talking about how life may have began on Earth and what kinds of the conditions it was like and how different it was today, and maybe you could find that on another planet or a moon. But I just wrote down. I found just some really interesting things about the conditions in which these critters can be found. They can be found in areas where the radiation is outrageous, 3 million rads. If you know anything about radiation, that's 3,000 times what would kill you. They can live in stuff that has a pH that's just incredibly acidic. Look at that, 0.03. This is conditions that the acid actually is capable of dissolving metal. Boiling conditions, right? Remember, 100 degrees is boiling in Celsius, right? The ocean, if you wanted, just for comparison, they can live in conditions that are greater than a 25%, greater than a quarter salinity. Just for comparison, the ocean is 3.5. So what? 8, 9 times more salinity than the ocean they can live in. And if you remember back from Bio 1, you ought to be thinking right away, wow, that's really hypertonic, right? If the ocean could be hypertonic to ourselves, and it is, right, then these kinds of things have to deal with just incredible environments. That's what's so cool about them, though, because they are what might be, before I start any alien conspiracy, but that's an interesting halo around there. They might be what were the distant descendants of something that may have come, like if you think about the panspermia thing that I was talking about, right? And the reason why we say that is because of the fact that they live in these just outrageous conditions. And when we see organisms that can do that, evolution would drive you to only remember. Evolution doesn't make you perfect; it only makes you good enough for the time. Well, when was it necessary to be able to live in a condition that acidic wise that can dissolve metal, right? Or 9 times saltier than the ocean. So that's the neat thing about it. We call these things extremophiles, and there's another list. Like they're extremophiles in acidity, alkalinity, temperatures. Some of them were hyperthermophile to love, right? Thermotemperature. Can live in temperatures beyond boiling, right? Psychophiles, really cold ones, psychro, that's what those are. Yeah, look at that, temperatures that would freeze anything. Halophiles love salt. Osmophiles, which live in high sugar concentrations. So these things can live in practically everywhere. I mean, they live in the tissues of other organisms, and that's kind of what you're seeing down below there. I can't remember what those are from the book, but that looks like the bacteria. They've got a much better view of it, if I'm speaking wrong. The one on the right of what you might see in a yogurt culture. These things are ubiquitous. They're found near those geysers, like near Yellowstone. See in the upper right? That's one of those where the water is like super hot and full of minerals and just really nasty. Or remember those geothermal vents too, right? That picture on the left, that's an interesting story. Back when I was first teaching this course, which is a long time ago, we found a rock in Antarctica that was a meteorite, and we had traced it to, we believe it came off the surface of Mars. Now, I'm not a geologist, but you just have to take the word of the geologist, the expert geologist, that this was a Martian rock. And if you're wondering how the heck did it get on Earth, well, two things ran into each other. There'd be splattering, right? So maybe a rock from Mars got thrown off after an asteroid strike, right, or a comet strike, and drifted off. And most of that stuff would fall back on Mars, of course, but some of it may have gotten far enough up that it drifted off into space and drifted for millions of years and blah, blah, blah. Eventually captured by the Earth's gravitational pull, and there you go, meteorite, right? It happens. So anyway, this is an electron microscope of that Martian origin rock, right? We didn't get it from Mars, we got it from the South Pole, but we believe geologically that it came from something off Mars. And that little weird thing that you see growing in the middle is what got everybody's attention when this first was shown, because that almost looks like something put it together. And when we think of one of the characteristics of life, organization, when we say something that's organized amongst a bunch of stuff that doesn't look organized, right? I mean, you really kind of see just a bunch of lumpy stuff there. That piques our attention. Astrobiologists or biologists in general. Really small. There's a lot of prokaryotes, right, that we saw in the microscope, the rod-shaped ones that kind of look like that, don't they? Does that mean that was a fossilized prokaryote? They don't fossilize very well, for one thing. No, because there are also some geological explanations to what that is. In other words, some things that would happen in the rocks that would make it look that way that aren't anything. And that's why I always caution folks to be careful about those shows where they go, look at the way these stones, they must have been humans that put it together. You always got to wonder, did the geologist go, no, that happens naturally. I know those spacecraft that fly around Mars and take pictures every once in a while, but say, it's a face on Mars, and there's some psychological aspect to that we recognize familiar things in places, so we think it's a face, and it's not. Or the face of somebody on a grilled cheese sandwich one time I heard about. Oh, look at the way it's grilled. It looks like somebody. Anyway. What do we know about prokaryotes? So now we'll lump those domains, and again, I'll try to be cautious and remind you if we're switching between them. But prokaryotes in general have these characteristics, and I'm not going to spend a lot of time because most of them are pretty self-explanatory. Unicellular, they can form colonies, which is kind of interesting because one of my really things that I enjoy about studying biology is trying to figure out or trying to understand when single-celled things became multicellular things. I'm making a big jump. I'm talking about going from a bacteria to like you and I, right? And it didn't happen overnight. It's kind of like acceptations, like the eyes we were talking about. It must have happened in little steps. So even in prokaryotes, we start to see this idea of cells living together, or not multicellular, right? Okay, in colonies, even in things called biofilms, the stuff that gets all over your teeth that causes cavities is a film of bacteria. Biofilms, and even in some cases metabolic cooperation. So what we're getting are the really precursors. Now that doesn't mean these things are going to become like multicellular. Remember, they are what they are. But if we go back and we look at this, this shows us that, cells have this, desire is not the right word, but let me use it, to get together sometimes. And why do they do it? Because it makes them more efficient. Sometimes no. Some cells are like, no, we're good alone. But some do actually form colonies. So we'll talk more about multicellularity, to kind of work our way up. We really won't get to true multicellularity, till we get into the animals. So, interesting, right? All right, there it is. The blue stuff are bacteria that eat holes in your teeth. And what they do is they ferment sugar, right? And you all know what that makes, lactic acid. And acid does what? We saw these guys the other day, metabolic cooperation. So an anabina, the little blue-green ones, every once in a while out in front of the building I can find some live anabina, but I didn't this time. So on the fixed slides it doesn't show as well. But that string of little cells there, those are all individuals, if you will. They live together in this group because guess what? There's the photosynthetic cells, and then there are these things that are called heterocysts. And heterocysts do something called nitrogen fixation. So very quickly, because we're going to go through this a little bit later on, plants need nitrogen to survive. So it's in fertilizer. Okay, so we'll come back to that too. Carbon fixation, I'm hoping you're not, I was going to say hoping you're not fixated on. I'm hoping you're not wondering about. That's what photosynthesis does, right? In chemistry, fix means to take something and like carbon or nitrogen and turn it into another form. Like plants take carbon dioxide, right? Turn it into sugar. That's what you mean by that. So there's this metabolic cooperation in the anabina where all of these cells need energy, and all of these cells, or food, excuse me, all these cells need food. All these cells need nitrogen. So there are some that specialize in making nitrogen, and the photosynthetic ones make enough food to feed them, and the heterocysts, make enough nitrogen from the cells. Right? It all need together happen. Yay! Different shapes. Right? I wrote this up on the board. Diplo, it's in pairs. Strepto in a chain. Staph is a group, like cluster in the upper right. If you're thinking about gram-positive and negative, I'll get to that in just a second. But those look like they may have been staying with gram stains. So if that's the case, then you do have some gram-positive and negative in it. The rod's a rod shaped and the spiral. Look at that one in the bottom. That cool picture I found of one standing up. See that little photograph? Not the stained ones. That's a helical. Helical means a spring is a helix. DNA is a double helix. I can believe I forget DNA. So the shapes, we talked about that. Sizes, of course, they're not very big. 1 to 5 microns. So one of those little anabina cells that you looked at under the microscope at 40x, right? One of those is about 5 microns. So that's about the upper limit of most of them. Versus much larger eukaryotic. Although there are some big prokaryotic cells, like that one you see there, Thiomargarita nambiensis. It's got all kinds of cool stuff in that word. Thio means sulfur. Nambiensis is a place in Africa. It's probably where they was first described from, or something like that. That second word can be kind of anything you want. Oftentimes it describes where the species was described from. It's not like it's infecting the bee. They put it on the bee to kind of show you a scale. That's good size. That's large. So that prokaryote must have ways to deal with being really big, right? Cells don't usually want to be really big. Now, with that Thiomargarita may not live in colonies. Although it might. I'm not sure. Okay, cell walls, surface structures of prokaryotic cells. 3 things that they do. Some of these actually... Oh, there we go. If you remember back to plant cell walls... Now, cell wall of a plant, cell wall of a prokaryote are different. Okay, functions, difference. You ought to be thinking, oh, this is all convergent evolution in a very, really big grand scale. But these are the three things that a cell wall does, like in plant cells. Cell shape, remember plant cells had that shape. Physical protection, okay, so they can provide... What cell walls of bacteria can do is they can prevent things from getting in, just like any cell wall can, to attack the bacterium, like the bacterium were like infecting something. Their cell wall can also use them to be incognito, right? I'll talk about that in just a second. And lysing, remember that's the big thing about plant cells. That's why plants like to be in hypotonic environments. They like having a lot of water in their water refuge, right? Hypotonic. The plasma membrane is a little bit different, and this is a picture that I got a little more in it than I need, but all right. Basically, if we look at bacteria and eukaryotic lipids, and probably the best place to start then might be down in the bottom left picture, the one in the lower left. You see the phospholipids. I hope you recognize those, right? It's phospholipids with the fatty acid tails facing in. So that's how the membrane looks in eukaryotes, like you and I. It's also the way it looks in one of the prokaryotes, one of the groups of prokaryotes, one of the domains, the domain bacteria. So us and bacteria have very similar plasma cell membrane structure. And then up above, there's a little bit bigger picture of those phospholipids, right? Don't get too caught up in the chemistry again. These are those molecules that are amphipathic, right? They ambivalent. They are dissolved in water and not dissolved. So that's why they form those bubbles. That's why they form cell membranes the way they do. But don't worry about what that fentanyl side chain, I don't think that's fentanyl, but phytenol, pretty close to it. But what I really want you to look at is something called the ester and ether linkage between them. If we look at the phospholipids that are in the archaeans, the one up above, that part in the yellow is an ether linkage. Okay, so that has to do with chemistry. I'm not trying to throw chemistry at you, but I want you to understand that what links those together, you can see in the picture the yellow things are different. The phospholipids from the bacteria and eukarya have carbons and double bonded oxygens, a little bit more complex, but that's an ester linkage, not named after your aunt, but it's an ester. Now, if you've taken chemistry or will take chemistry, you'll become more familiar or are familiar with these linkages. I don't really care if you can discern an ether from an ester. Here's what I want you to know. In archaean phospholipids, right, the linkage is an ether linkage, so I don't care if you know ether, but I want you to know in the red, it's more resistant to hydrolysis. If you have ether linkages, so the actual phospholipid molecule in an archaean, domain archaean, has a slightly different linkage, which makes it more resistant to hydrolysis. Well, I lied to you before and I said I didn't want to worry about the fontanel side chain. Again, don't worry about the name, but notice that because of its chemical makeup, there are things sticking out of it. Those little things are representing bonds. We didn't draw all the atoms in. Remember some of the shorthand that we use in organic chemistry. So when we now I'll go down to the bottom, compare the phospholipids from the bacteria and eukaryotes, and look at the ones from the archaeans. Not only do they have a linkage that resists hydrolysis, their phospholipids are connected to each other across the membrane and down the membrane. Those little things that stick out make them stick together. It's like a super glue of cell membrane. Excuse me. So we don't talk about a bilayer of phospholipids in archaeans. We talk about a monolayer. All of this that I've been going through is just to show you that archaic means ancient. And the reason why we gave that name to a lot of archaeans is they represent, again, some of the maybe descendants of some of the early critters that we found on the planet, because they have these weird things. Again, it's a detective work in biology. If you find critters with this kind of a membrane, you go, why do that? Why do something if you don't need to? Because that's a lot of energy and materials. So later on, when things got a little bit easier on Earth, maybe, then it was okay to make that phospholipid, and there would be an advantage to that too. Maybe you could be more flexible that way. I could see bacteria and eukaryote membranes being more flexible in archaeans, right? So it's a give and take. Now, the plasma membrane, not only in terms of the phospholipids, but in terms of the layers, is different too. And so now we're going to be dealing with just domain bacteria. Okay, so we're not going to be dealing with archaeans here. So they've got their membrane that's different than the ones that the bacteria have. But now amongst the bacteria, amongst domain bacteria, there are the gram-positives and gram-negatives. Now, if you've taken micro, you know what that means. When you stain them, you look down below. There you go, right? Gram-positives are purple, gram-negatives are pinkish, not purple. There's a reason behind that. And I talked about in lab the fact that this is an important thing to do in micro, because with micro we identify microbial organisms, among other ways, by how they react to stuff, stains, right? And the way they react to a gram stain has to do with the difference in their structure. Gram-positive bacteria have the plasma membrane, so you should recognize that. That's the one on the left with the phospholipids. But there's a layer of a peptidoglycan. It's a protein sugar layer. That's the cell wall on the outside, right? So cell wall, but not cellulose. Okay, so don't confuse those. On the right-hand side is a gram-negative bacteria. You notice that its cell membrane wall structure is a lot more complex. For one thing, it's got 3 layers, doesn't it? It's got an inner plasma membrane layer. It's got an outer plasma membrane layer. Then it's got a thin layer of peptidoglycan in between. I want you to focus on the thick and the positive, and the thin and the negative. And also keep in mind that in the negative, we've got a double layer. Now, those little green things that are sticking out are lipopolysaccharides and lipoproteins. In other words, they're fats that are coming off of sugars and proteins. You probably studied those in Bio 1, at least I hope you did, because you were talking about cells being able to recognize one another. That's how immunity works, right? So I mentioned classic gram-negative bacteria. One of the reasons that interests a clinician would be that many of our diseases that are bacterial in origin, or not all, but many of them tend to be gram-negative. So you would do a very simple stain. You can do it in micro. You did if you took micro, and find out whether it's a gram-negative bacteria. But the reason why that happens, the reason why you get the different colored stains, says something about the structure of the membrane too. In gram staining, and we don't do gram staining because you learn it in micro, but what I want you to know, in gram staining, you stain the bacteria purple first, right? I think it's crystal violet, so it doesn't matter, right? The next thing you do is important. You wash your bacteria, your organisms, you wash them in alcohol. What alcohol does is it makes the peptidoglycan layer shrink. That's why I want you to focus on thickness. But notice in the gram-positive, there's a lot of peptidoglycan. So think about shrink wrapping something. And think about in the gram-positive, you're shrink wrapping with thick plastic. I think I've actually seen things that are shrink wrapped in something a little bit more durable, right? And then on the right-hand side, the gram-negatives have a very thin peptidoglycan layer. So it shrinks with the alcohol. Alcohol dehydrates it, okay? Now, here's the important part. The alcohol, once it shrinks to the peptidoglycan layer, washes away the purple stain. But, this is important, because the gram-positive had a thicker shrink wrap, had a thicker peptidoglycan, it traps the stain. So the stain gets trapped inside of the bacterium. So when the alcohol shrinks the layer, it can't remove the stain. But those that have a very thin layer, alcohol had no problem. Washes away that layer, washes away the stain, and those bacteria then are clear. So how do they get red? Well, if you remember doing a gram stain, and if you don't worry, you come back and you stain them red later on. You stain them with saffron red. The reason why you do that is even the purple ones take up the red. All of them take up the red. But since the purple ones are so darkly purple, the red gets masked. And you put the red on just so you can see the gram negatives, right? So there is a physical reason as to why gram-positive bacteria become gram-positive or purple. They're trapping the crystal. It is crystal violet. You just said it right there. They trap the crystal violet, and then later on, the crystal violet gets washed away with the alcohol. It's not written exactly in the right order. Crystal violet is easily rinsed away, revealing the red saffron dye. It's actually more a matter of you go back later on and dye them after you've rinsed all the dye away. So, either way, right, it's the cell wall that allows you to have the difference, and the difference is that peptidoglycan layer. Now, we don't have a peptidoglycan layer, neither do archaeans. Right? We don't have that layer on the outside. None of us eukaryotes have that. So look at this big picture here, right? Gram-positive, gram-negative everywhere. And if we were in class, but I bet you it's obvious, you can see that those are, for the most part, round ones. There might be a few rods in there. Some streptococci and staphylococci, a whole host of them. Let's stick with that cell wall. Here's one thing that they can do, these bacterial cells. They can invade your tonsils That's a tonsil cell there on the right. And around the outside of that cell wall can secrete a capsule, a slime layer, right? It could be thick, it could be thin, but basically what it does is that, number one, this is Streptococcus right here, for example. This is the thing that causes strep throat. There it is, that organism right there. That cell wall having that slime layer around it, number one, allows it to stick to your tonsils, which is good because that's what it wants to do. That's what it wants to infest or infect. Helps from drying out, so that would be important in those kinds of conditions that are hypertonic. And it hides you from the immune system. Remember all those things that stick out of the membrane, the lipopolysaccharides and lipoproteins? I mean, that's not just in prokaryotic cells. You have them too, but that marks the cell. It gives you identity. So if we hide and the immune system comes along in your body, which all it really does is check those and make sure those structures are correct, which are decided genetically. This is reminding y'all. But it's covered in slime, right? So I can't see it. So I'm hiding incognito. That's what I mean by that. So that would be helpful if you have one of those slime layers. And you get in, you infest the tonsils. I mean, if it was just one streptococcus, your immune system might be able to take care of it. But since you can't recognize it, they grow. Cell walls can also collapse down and then be shed off and eventually form this really thick wall called a thick wall that surrounds something called an endospore. And this organism here can make endospores that can last a really, 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 really long time, sometimes on the order of centuries. This is Bacillus anthracis. There's a name in the upper left. This causes anthrax. Anthrax spores can last for decades, I believe, maybe even 100 years. Some cases of bacterium, they can even last longer. Remember in evolution we talked about the fact if the environment changes, right, that's gonna cause organisms to have to adapt, or they can go extinct, which is what they usually do. But there is another option, and I may have hinted to it before, or maybe we spoke about it, I don't remember, that you can just kind of wait things out, right? You could form a spore like this bacterium is doing. And if the environment's not good for you right now, you go into a spore, so you kind of go into a hibernation type of thing. I don't really like to use that word because it's not relevant, but you get the idea. And then if conditions are right sometime in the future, maybe decades later, you can then germinate. What does an anthrax spore need? Well, moist, warm locations are good for it, so getting it up in your nose is not a good idea. If you remember years ago, you might not, some of you weren't alive then, but right after 9/11, there was an anthrax scare because somebody was sending anthrax spores through the mail. It looks like the powder white, I guess, and so folks were, in some cases, were being sent anthrax. For reasons that escape me because it was so long ago, but it was just all part of that weird time back then. Extensions off the cell wall, these things called fimbriae. For the one in the lower left, what they can do with those fimbriae, they can adhere. Adhere is sticking to something different than yourself, right? So it's not, if you, the bacteria aren't sticking to other bacteria. Or of course they could with that. I guess I should probably modify that in that case. But more importantly, they can stick to their host cells. They can grow these fimbriae out and actually form something called a sex pili, which is interesting because these organisms don't have sex, but they do exchange genetic information. So that is by definition sexual reproduction on some level. But you can see what's being exchanged there, right? And we'll talk about that later on. TEM, if you're wondering, is a transmission electron microscope. If you wonder what's being exchanged there, if you look at the organism on the right, they have these fimbriae that are sticking out everywhere. The organism on the left, excuse me. The one on the right is not, right? It's naked. Now, if those fimbriae give an advantage, like being able to hold on to the host cell, then that would be something that the cell on the right would want. And bacteria, prokaryotes, have this really unique ability to, like, within a species and sometimes between species. For the one on the right, if you could think about a conversation, the one on the right says, hey, those fimbriae really help you hold on. And the one on the left says, you want them? And the one on the right says, sure, I'll take them. So I can give you a copy of how you can make them. And it gives it some DNA. Gives a little piece of DNA, and that DNA, that's the information about how to make fimbriae. So that'd be kind of cool, right? If we could go to a bird and say, hey, can I get those wings from you? Yeah, here's some DNA. Check them out. You got to be relatively closely related, don't you? You think The flagella are different, really, really, really different. If you look in the upper right, there's a picture that shows the difference in the flagella between prokaryotes and eukaryotes. You remember eukaryotes with these microtubules that slide by each other? That's what makes the flagella wave. Right? But in a prokaryote, it's actually a rotor. It spins. So this is a really cool example of convergent evolution in it, at a really grand level. Got the same thing, does the same thing, moves the critter around, but it does it really different ways. I want you to focus down in the lower left on the red where I wrote exaptation. It's a good review of what we talked about in class, right? Exaptations are genetic adaptations that end up becoming fortuitous later on. The eye. Remember, we talked about the camera eye. Your very specialized eye. Stephen Jay Gould was right. Or he raised the question in his rhetorical statement of what good is 5% of an eye? The eye that we have now is no good if you only have a part of it. You got to have all of it. So how did that come about? And we talked about the little steps along the way and the fact that we have evidence for that, right? With the limpets and the squid and that sort of stuff. So on exaptation, the reason why I put that in red is the way that rotor spins is hydrogen ions flow from one side of the membrane to the other. So the direction you see in the arrow, hydrogen ions go from, so this is obviously what we're, a gram-negative bacterium, isn't it, right? Because it's got two of those membranes. So the hydrogen ions flowing from one side of the membrane to the other cause that flagella to spin. There's a really cool picture on the right, under an electron microscope. This is so neat. It's like miniature technology. Anyway, that makes it spin. And if you're wondering, I still don't get the exaptation, go back and review respiration, or even photosynthesis. Go back and review ATP synthase. Remember, that's what spun that too. That's what spun ATP synthase. All right, exaptation in big scales, convergent evolution in big scales. How about reproduction and genetic variability? So mutations can arise, radiation does it, chemicals does it, but you know how that is, right, with mutations. They can arise spontaneously, so we change a T to a G or something like that. You know, and that's not something that Mother Nature is really, she's like, okay, you can try it if you want, but you're probably not going to like it. Remember, that's why even the code table is built with that wobbly base, remember? Just make sure that even if you mess up the last base, in many cases, you still get the same protein. As long as you get that, you get the same structure, and that's the whole point anyway. But they can reproduce a lot, remember? So that helps. Go back and review that. They can make lots of offspring, so they can get away with the fact that most of those mutations aren't going to be good. They don't have meiosis, so they don't have the ability to do what we call vertical gene transfer. That's what you and I do. The organisms that go through meiosis, animals, in plants and fungi and that sort of stuff, what we do through that process is we vertically transfer our genes. We pass them down to our offspring. If you remember binary fission from Bio 1, the bacteria, it makes a copy of the single genome, the single gene that it has, makes a copy, then it splits in half. It's very simple, binary fission. So that's horizontal gene transfer. So you make offspring, yes, but you make offspring in the horizontal fashion, if you understand what I mean by that, right? There's other ways they can get genetic variability, though. There's other ways, and this is probably some of the bigger ways in which that happens. They can absorb a fragment of DNA from the environment, either from other prokaryotic species they can. That's the picture I'm showing you, the first one. The two cells in brown, the little green thing is a piece of DNA in the environment that bacterial cell says, I wonder what this does. Takes it up, plugs it into its chromosome, and now it makes that stuff. Maybe it's like, that wasn't good, and the cell dies, or maybe it gives an advantage, right? Even from other prokaryotic species. Very cool. Viruses, I'm going to come back to this in just a second, so don't worry. Viruses can act as a vector. That little green thing with the looks like a stop sign is a virus. Viruses aren't really alive or not alive. They're a weird thing. There's a chapter in your book. I don't go over it, but not because I don't consider them important, but they're, we got a lot of other things to talk about. Here's a cool picture. That's what the virus in the upper right is. What a bacteriophage. To feed upon bacteria, bacteriophage. It's like a spaceship. Basically what viruses do, and then down below is one cell being attacked by a bunch of bacteriophages. Viruses, what they do is they inject their DNA. That's what that blue coiled stuff is up there in the head of the bacteriophage. They inject it into the host cell. It gets into the DNA, and then the host cell, while it reads through its DNA, right, its transcription, it unwittingly, that's not the right word, but you get it, but it makes viral DNA products. And what's the viral DNA products? More viruses. So it makes more of the caps and the legs that you see. It makes more of the things to make more viruses, and then when it makes enough of those, then the cell lyses, out comes more viruses, and then the cycle repeats, right? Well, that can be good or bad. The bacteriophage is not doing it to help the cell, but maybe it interjects a piece of DNA that for some reason the bacterial cell or the prokaryotic cell can use for something. You know, that could be some sort of genetic variability. You remember, we're dealing with cases that are the rarity, but they happen. And then finally, that was the one, the sex pillai. So there's what I'm talking about. The little green piece. Many bacteria have an accessory piece of DNA. In the picture down below, the purple ring is their gene. They're one chromosome, not their gene. There's several genes. But in the green, in some bacteria, like for example, the one that causes tetanus, they have a little accessory, a plasmid, right? And what happens is the plasmid can have genetic information that gives the cell a specialty of some type. So let's imagine in the picture on the right, the plasmid in the one on the bottom, which the one on the top does not have but is getting, gives the ability to make those fimbriae. So it's another piece of DNA. It could have been something they picked up from somewhere, to be honest with you. But the cells then get together, a sex pillai forms, and the one down below, then you see in the picture, transfers that plasmid, that accessory piece of DNA, to the other one. So if the one on the top says, hey, I'd like to need those fimbriae because that seems to help you attack this host cell, the one down below says, well, I tell you what, let me give you, and it doesn't give it to it, but it gives it a copy. So that green circle, you notice the circle is staying in the one on the left, but the copy is moving to the one on the right. So, very cool, right? So it's kind of unconventional, but it still gives you genetic variability. But it's going to really depend on numbers, isn't it? You're going to need to make sure that this happens a lot, these random kind of incorporations of DNA and mutations. But they can deal with that. They make a lot of offspring. Okay, metabolism. I got 2 slides on metabolism. This one is cool to look at, and it's interesting, right? It says the same thing as the other one. The one I want you to focus on, though, before you go, oh my God, is this one. Metabolism in general, and this is out of your book, is what the type of metabolism you engage in is dependent upon 2 things. And I put it in the light blue box down in the middle, right below where it says table 22.3. That's the one. It's the type you are depends on where you get your energy and where you get your carbon. And then at the very end of that, we add the word troph. That means to feed. So the kind of feeder you are. So let's kind of start with a simple one first. Now, let's go with the picture down below. Let's say your carbon source is organic compounds, food. Okay? You're a hetero. All right? Because you get your carbon from someplace else. You get it from an organic compound. You're going to see what I mean, the differences in just a second. The energy that you get is either from the light, so you're doing it kind of like a plant, but you're not a photosynthesizer, you're a photoheterotroph, and only prokaryotes can do that. And I point that out because that's weird to be able to do that, which means they can live in weird environments. Or you oxidize the carbon that you bring in. You digest. You go through respiration. That's where we would fit. So we would be a chemoheterotroph, okay? On the left-hand side are the plants. We'll go with those first, right? Their carbon sources from carbon dioxide, so we call them auto. They didn't make their own carbon, but they did use an organic compound to get it. See, when you eat food, when you eat sugar, the carbon and the molecule, there's 2 things that are going on there. You're getting the energy from the breaking of the bond of the glucose molecule to carry on life and ATP and all that sort of stuff. And you're getting the carbon to use as a foundation. Remember, that's why it's a foundational element, to make other things, to grow, to repair, to do the things you need to do metabolism. Plants, on the other hand, don't get their carbon. They make their own food. You know that. So they get their carbon from an inorganic molecule, carbon dioxide. And then their energy source is either light on the left, that would be plants. So plants would be photoautotrophs. And then there are some out there, again, it's only prokaryotes, that can oxidize inorganic materials. So these are the ones that can do things like nitrify bacteria. These are the ones that are important that I talked about in the heterocyst with the Anabina. Plants need nitrogen, right? But they can't use the nitrogen that's in the air, even though the air and the nitrogen in the air, the nitrogen in the air is about 80%. They can't use atmospheric nitrogen. So where do plants get the nitrogen? They get it from the soil, and they get it from the bacteria that live in the soil, because there are some bacteria, chemolithotrophs, that can make their living off of using those nitrogen compounds and using atmospheric nitrogen. We'll talk about that when we get into the ecology here in just a second. Up above, you notice that the names get a little bit, like there's the chemolithotrophs. There's also another group, they call them the chemoorganotrophs. So this is, there are just different names for these. I'm satisfied if you know auto and hetero, photo and chemo. The ones down below, I'm satisfied you know those names. Really the one up above, what I want to demonstrate is that look at all the different ways organisms can make food. Now this includes the prokaryotes and the eukaryotes, because here we are over on the right, organic heterotrophs, autotrophs, right? On the left is going to be a lot of these organisms that do it really weird, or critters like the prokaryotes. So down here in the lower right, I showed you this picture earlier of the Titanic. There it is, Halomonas titensia. This thing oxidizes iron. Fe is iron. It oxidizes iron, so it gets energy from that. Then it oxidizes organic compounds in the environment, and it gets energy from that. So it's kind of like us, except there's a step before. See the dude up above eating a burger? That's you. You are oxidizing the carbon compound to get energy from it. This organism that's living on the Titanic's iron is oxidizing the iron and then using the energy from that to oxidize the organic compound. So step before. Some are also, they oxidize not just iron, they can oxidize sulfur. These are some of the organisms that we talked about that can live in those extreme environments, like down around those geothermal vents. So this is more for curiosity. I'm not going to quiz you on the differences here, because the same thing there is there. And again, I'd like you to focus on down below, naming the different types of ways organisms can digest food. Oh yeah, ecology, you got to talk about those. Ecology, of course, we always like to talk a little bit about how these organisms interact with their environment. For one thing, they're photosynthesizers. We talked about that in lab, Anabina and Oscillatoria. All those cyanobacteria, man, they were so important because they're the ones that changed the gas composition on Earth, right? Remember, it was reducing. These guys made oxygen. They didn't make oxygen. You remember photosynthesis? You don't make oxygen. The purpose of photosynthesis is not to make oxygen. Oxygen is just the leftover product from the water, right? Photosynthesis, first thing you do is you split the water molecule, right? So the oxygen is just kind of a byproduct. Now the plants use some of that for respiration, but they don't use any of it for photosynthesis. It's a byproduct, but it floats in the atmosphere, and then you can start engaging in aerobic respiration. Carbon cyclers, they decompose things. Lignin, the name of that molecule with that really big, very complex structure, the picture up above it is what lignin is. Lignin is a component of the cell wall in addition to cellulose, which is tough enough anyway. But lignin is one of the hardest substances on the planet to break down. It's important for plants for protection and being able to stand up, but when a plant does die, a tree with a lot of lignin and it dies, all those little carbon atoms you see in there, even the ones in the circle, if you remember the way that shorthand works, all those, if something didn't decompose those would be lost to the environment. So prokaryotes are very good at providing that. They break down these molecules. They separate up those carbon, putting them back into the environment. Now they're doing it for their own, right? But along the way they make, it's like plants, they make more than they need, right, if you will. So they increase the carbon concentration in the environment. And that's important because other organisms need that carbon and they can't decompose it. So that's the ecological part. We talked about the nitrogen, right? We'll talk about that again in just a second. So they're nitrogen fixators. They take nitrogen from the air and they incorporate it into some chemical compounds that plants can use. Now, you might supplement that with fertilizers, and if you go to the store and you look at the fertilizer, it'll tell you the nitrogen concentration on it. That's those numbers, those 3 numbers you see with the fertilizer. So just a real quick, this is probably something you drew a cartoon or maybe you colored in the 6th grade, the carbon cycle. We have to cycle carbon, right? Carbon dioxide is given off into the atmosphere from a variety of sources, including us. Other organisms photosynthesize and take that carbon and put it back into sources that other plants can use. Remember, the glucose that you eat, you get the energy from the breaking of the bonds, and you use the carbon in there as building blocks to make your things. Right? So that's all part of the carbon cycle. I'm not going to ask you specific questions on the carbon cycle. Know that prokaryotes are obviously important in that. Here's the nitrogen one. So a process of nitrification, ammonification, and again, this is not a chemistry class, but what I want you to notice, the arrow on the left, nitrogen in the air, atmosphere, plants can't use that. In the bacteria, and many oftentimes found growing on the roots of certain plants like beans, legumes have these nodules growing on them, and what those nodules are colonies of these bacteria, these nitrogen fixing bacteria. So of course the nodules on the plant's not doing it for anything else other than itself, so it's concentrating these bacteria, meaning these bacteria are fixing nitrogen, doing their thing. They're getting energy off of it, but they're also turning nitrogen into something that the plant can now use. Well, of course, in the nodules, they fix more nitrogen than the plant needs, and so the soil is the benefactor of all that, and the soil becomes richer in nitrogen. Decomposers give off, when decomposition or urination, that sort of stuff, that is an ammonification that occurs, and of course, that causes the soil to become very basic, so there are bacteria that take care of that. Right, so there are bacteria that are very important in what we call the nitrogen cycling, and there are even bacteria that denitrify bacteria. So they're all important in keeping the nitrogen balance perfect in the environment. If you take ecology, or even if you don't, I can tell you now, that we often talk about in plant growth there's a limiting factor. Sometimes it's light, Very often the limiting factor is nitrogen. The amount of nitrogen in the soil is going to be the limiting factor in a plant's growth. More nitrogen, more growth, less nitrogen, right? Less growth. It's important, especially for a plant. Let's talk about symbiotic relationships for just a second. Symbiotic living side by side. Now, this is something that's going to span through several semesters, but it does start here with the prokaryotes, so I do need to talk about it. And I made yourself a nice little chart. There are basically 5 types of symbiotic relationships. Ecology is organisms living together. Not necessarily just in the same species, but separate species or maybe even separate domains, right? Bacteria live in your gut, so there's a symbiotic relationship. And symbiotic relationships are 2 species living together, but they're not always 2 species living together to help each other out. So let's look at some examples. Parasitism. In parasitism, one organism gains the benefit. I'm sure you can guess who that is. And the other organism is harmed, right? The negative means negative benefit, if that makes any sense. And I hope you know who that is. That'd be the host, right? Predation is a weird one to put in there, but it's a symbiotic relationship, right? One of you gets eaten, one of you eats. So it's pretty obvious who's positive there. Commensalism is an interesting one. It's one that you drive around here in Florida, the white birds that follow the cows. Those are actually, many of those are egrets that flew over from Africa. It's an example of that founder effect stuff we talked about. That's an example of a commensalistic relationship. The cows are kicking up insects when they're walking around grazing, and the birds feed on the insects. So the birds gain a benefit, and the thing on the right, a null, means that the other species, the cow in this case, it's neither harm nor help. It doesn't care. The bird is taking advantage. Pilot fish. We used to think that the old story was pilot fish, the black and white fish. We don't have them here in the Atlantic, but in the Pacific, you always see associated with sharks. We called them pilot fish because at one time we thought maybe it steered the sharks to their food. but that's not true. They're a commensal organism. They hang out with the sharks. They're too small for the sharks to deal with, but they gain a benefit because when the sharks feast into something, they can feed upon the chunks, the little pieces, right? Mutualisms like Nemo or the the clownfish living in the anemone, they both gain a benefit from that. You scratch my back, I'll scratch your back. And the one at the bottom might be a little interesting. Competition, you think about, is that yeah, that's two organisms in a relationship to each other. Why is it negative for both? Well, if you're competing for something, you don't get 100% of it, right? If you're if you're getting 100% of it, there's nobody to compete with, so there's no relationship. So if you're in a situation where you and another organism are competing for a resource, you and another type of bird or whatever the case may be, you might get 80% of it, right? But you don't get 100. That's why we say it's negative on competition, okay? I'll show you more examples of that as we go through this group as well as some of the other groups. They're found in all the groups. The diversity of these organisms. So I've talked about this very briefly at the very beginning. There's the universal ancestor, and then we thought many years ago that it broke into 5 kingdoms, but now we know that that whole kingdom thing is kind of up on the edge, especially in the eukaryotes up above there. But we do know we have 3 domains. That's the big group, right? Is distinguished spelled wrong in that? It says it's got this. If distinguishes, I'm sorry. So, number of species is debated as well as what distinguishes them. These are really small things. Just putting them lumping together because they're small. There's a lot of animals. Well, they're not this small, but there's animals that are really, really small. We'll see those in the protozoan. So, just because you need a rotifer is an animal. It's not a. If you remember what rotifers are, if you don't, then you will. So, being small is not a reason to lump you together. Now, I've talked about some of the, conditions that separate these apart. The difference in the phospholipid membrane was a big one, right? Especially between the archaeans and the bacteria. What's really interesting about this slide, before I move on, though, is when I'm pointing to with the red arrow. Notice who our closest. That's a really, 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 really relative term. Relative is, though, right? Us domain eukaryotes. If you think about it, the prokaryote group that we're more closely related to, relatively speaking, are the archaeans, the extremophiles, than the bacteria. I don't know. That just kind of strikes me as interesting, right? That the more extreme, weird critters that live on the high radiations in very acidic environments, that's our closest relative. And I'm really using that word closest with some really big quote marks around it. You can see me using my fingers. Let's talk a little more about diversity. There's some things that separate the 3 groups. I would like you to know some of these, maybe all of these. Yeah, why not? Nuclear envelope, you already know that part. You should know that prokaryotes don't have one membrane enclosed organelle. So yeah, you can know these because you learned about them in Bio 1. Peptidoglycan, we talked about. The absent peptidoglycan in the archaean and eukarya, I would just say is, even though it's weird, because the membrane lipids are the same in bacteria and eukarya, because the absence of the peptidoglycan layer, cladogram folks, people who study this professionally, tell us that that's one of the reasons why they believe archaeans and eukaryans are more closely related than eukaryans and bacteria. So it's, even though the membrane lipids are the same in those two, it's really more about the peptidoglycan for some reason. You'd have to talk to a microbiologist about that. Bacteria don't usually have introns, right? So any mutation that occurs is going to be expressed, right? Introns and exons. Introns are the parts that are cut out. Here's another little bit of evidence that the detectives out there suggest the archaeans and the eukaryans are more closely related. The archaeans have some introns. We have many, of course. The chromosomes are the same in the prokaryotes. It's one singular circular chromosome. We have linear chromosomes, right? We don't have circles. And bacteria are kind of like us. They're more. There are some extreme living bacteria, but in terms of temperatures, the archaeans win hands down. Some of those can grow at temperatures not the rest of us can't grow at all. So let's just do a real kind of quick, long run through the diversity. Among the domain bacteria, there's a clade called a proteobacteria, and a clade is again a family group, right? it's only called a monophyletic clade. It's called a paraphyletic group or a polyphyletic group. If you said monophyletic group, I guess you wouldn't be wrong, but you would be kind of, missing the point of the monophyletic is the only true clade. So proteobacteria, by this definition, must have all had a universal common ancestor. And hopefully we're describing all of them. Remember, this thing changes, new discoveries are made. And they break the clade proteobacteria, that's the biggest clade in the domain bacteria, they break that down into these groups that they give Greek letters to, alpha, beta, gamma, delta, and epsilon. And I just jotted down a few quick notes about them, which I would like you to know. Amongst this group, these are some of the groups we think were the origins of the mitochondria. If you remember back in Bio 1, we talked about the endosymbiotic relationship. They also discuss it in your book, how we think that certain organelles, because they have their own DNA and they divide. Remember mitochondria and chloroplasts, right? If you don't remember, go back and look at your Bio 1. We think that maybe some of them were the origins of that. They're nitrogen fixers. The one on the right is a diplococcus, but it's one that hopefully you've never had experience with. But Neisseria gonorrhoeae is the one that causes gonorrhea. Sulfur bacteria, they oxidize sulfur. Members of this group can live in very bizarre environments. Sulfur bacteria would be ones you'd find in high concentrations down around those geothermal vents, right? A lot of sulfur coming up through the water there. Some of them form fruiting bodies during adverse conditions, like the one up above, the Myxobacterium, what is it, Chondromyces crocatus, right? That's the fruiting. Now I introduced the idea of a fruiting body. Mushrooms, when we talk about them, a toadstool is a fruiting body. A fruit, they don't have fruits. Only plants make fruits, right? But this is a reproductive structure. And notice that the reproductive structure with the little lumps up above, when we get into the fungi, there are some fungi that make reproductive structures that kind of look like that. As a matter of fact, a myxobacterium, means a fungus. So it's not a fungus, but it looks kind of like a fungus, convergent evolution, right? And the important part is these fruiting bodies in this particular bacteria during adverse conditions, and the reason why they're going to go into reproduction and make these fruiting bodies during adverse conditions is the environment is changing, and you're trying to crank out some genetic variability that'll help you survive, right? So you're doing a lot of reproduction, you're doing a lot of depending on mutations and all those other things we talked about earlier to get something that'll survive. Many of the members of this group are part of our intestinal microflora, right? Our bacteria that live in our gut. And interesting enough, a few years ago, they discovered one called Heliobacter pylori, and it was the source of a lot of folks' stomach issues, like ulcers. And they found ulcers are caused by many things, but one type was caused by an infection of this bacteria. So simply take an antibiotic, helping those people that for years were dealing with, thought they were lactose intolerant or things like that. And of course, our gut is full of microbacteria. Yeah, it's very uneasy to talk about it. I thought this is kind of cool. Delovibrio is predatory. Pseudomonas is a bacterium. Delovibrio in the upper right with its big flagella. It can move fast. It can spin that sucker so fast it can move, what I say, about, scale-wise, 220 miles an hour, 100 microns a second. And then as it attaches to the Pseudomonas, it spins that flagella and drills a hole through, spinning at revolutions of 100 revolutions a second as it builds into its prey. So you think life is tough up here in the multicellular state or road? Think about that. Think about this. We don't say the B, so it's Delovibrio, predatory bacteria. It's a dog-eat-dog world, even at the bacterial level. Within this bacterial domain, there are the chlamydias, and I'm sure we've all heard about chlamydia. Chlamydia is a symbiont. These are obligate intracellular parasites. In other words, obligate means you have to live as a parasite. There are some organisms that can live as a parasite and not as a parasite sometimes, but they would, what's the opposite of it? Opportunistic, maybe, parasites? Inobligate? I don't know. But obligate parasite is you have to live inside a host. So that means chlamydia spread from host to host, right? And we all understand all that. Chlamydia trachomatis is the most common cause of blindness, and worldwide the rates of chlamydia have skyrocketed. If you look at the graph on the right, since the 1980s, and I don't know what the recent numbers, but chlamydia skyrocketed. And the reason why it is I guess it's easily treatable for one thing, so it's not a death sentence like syphilis would be necessarily, but it's a silent sexually transmitted disease, STD. Silent diseases are diseases that are passed, they're diseases that give you no symptoms. So you don't know you have chlamydia. And that's how it gets spread, right? You see the picture on the right now. Syphilis has dropped considerably. Gonorrhea has dropped considerably. Those are all very symptomatic things, but chlamydia, you can have chlamydia and carry it around and not know it. Meanwhile, having sex and, spreading it around. That's why you always got to make sure you use right protection if you're not sure of the environment, right? Spirochetes are those that live helical lives, and there's many free living, but some are parasitic. The one that causes syphilis, that little picture on the left is the one that causes syphilis, Treponema pallidum. Syphilis is one of those hardcore types of sexually transmitted diseases. It can appear, and then it can appear to go away, and then it comes back. It's really, in later stages, it can cause things like psychological issues, problems. For example, Adolf Hitler. They have some videos of him later in his life where he's got these constant shakes. Some folks suggest maybe Parkinson's disease. Some folks suggest maybe the later stages of syphilis. That's what happens. You get syphilis as a young person, and it comes back later on in your life, at least dormant for a while. Borrelia burgdorferi is the one, the Lyme tick disease. Lyme tick disease is more prevalent in the upper New England area for concentration, the Midwest, because the concentrations of deer. We do have concentrations down here, and there's what a deer tick looks like versus a dog tick. Deer ticks are much smaller. I've seen a few. I've seen mostly dog ticks. The critter Borrelia burgdorferi is in the lower right, the blue. It's a spirochete, and if you get bit by a deer tick, the organism causes that sort of target color or target presentation you see on the inside of the arm. Can't really tell what kind of body part it is, but if you have something on your skin that looks like that, you probably ought to be going to the doctor. That's a weird looking thing. Even if you had that little red mark there in the middle without the other one, I'd probably be off to the doctor. In this group of bacteria, the cyanobacteria, we love them, we praise them, thank you, thank you, thank you. We tell all of them why, because they made algae, right? And some of them actually may have been the origin of chloroplasts. Remember, chloroplasts gave the ability for these cells that didn't have it before to photosynthesize. Now, we love these algae, and we're happy for what they did, but they can get a little carried away. See the picture on the right? The lake? That little lake off to the right is obviously kind of a clear-looking lake, and the other one looks more like pea green soup. That's because algae, if they're found the right conditions, and nitrogen is one of them, and nitrogen's in fertilizer. Many times lakes that are around golf courses have issues with this because they use a lot of fertilizer to keep that grass real pretty on the golf course. If they use too much, it runs off into the lakes, and all that fertilizer then gets the algae to go crazy, and they start growing and growing and growing and growing and growing. And you think that's great because the fish got a lot to eat on, right? Algae also respire. So while they're growing like that, they're growing so fast, it's actually caused the oxygen level in that lake that's pea green there to drop, and fish will start to suffocate. They'll literally some species will start coming to the surface. You can see them sometimes, especially in the summer, because warm water is the same way. They'll gulp air at the surface, and that's usually because they're living in these conditions. Totally quicker. So good and bad, right? Now, let's stop for a second. Algae. The word algae is confusing because there are blue-green algae, and those are prokaryotes. Okay. There are green algae, and they can be plants, or they can be protozoans. Then there are brown algae. They're protozoans. So I will try to remind you, but don't get an algae is a filamentous type thing, and so it's more of a physical description word than it is of a biological related word. Okay, so there are cyanobacteria called blue-green algae because Anabaena has kind of a blue-green color to it sometimes. The gram-positives, and I didn't say it wasn't always just the gram-negatives, but two of the gram-positives that we really deal with are Streptococcus pneumoniae, Streptococcus pyogenes, They both lack that outer membrane. They're soil-dwelling decomposers. That's nice. That's the good. But there are some bad ones. There's the anthrax, botulism. That's a gram-positive. That forces botulism is one of the ones that can make those endospores. Look how toxic it is. 1 gram can kill. Is that a human? A million humans, right? Clostridium botulism. From what I understand, death is pretty quick, within a matter of a few seconds. Yeah, and it is a real heavy-duty type of food poisoning. It can be found other places, but it can also be found in food. Very rarely, though, so you don't need to worry about it. I wouldn't worry about it. Staphylococcus, streptococcus, all of those. Many of those are implicated in pneumonia and strep throat. You remember Griffith's work in Bio 1 was with streptococcus pneumonia, the one with the mice, and some died and some didn't die. Now, to the domain Archaea, very quickly, because it's a neat group, because they're very extreme. If you ever walk around the swamps here in Florida, you might smell methane, sort of that rotting egg smell. There are organisms that can actually live on those methane. They actually can use that. There are bacteria that produce the methane as a byproduct, or the methane is sometimes a naturally occurring product of decomposition, right? Or just a naturally occurring gas, because there's methane on, it could be life there, but there's methane in some of the moons around Saturn and such. But there are some critters called methanogens, or archaeans, that actually can digest methane. Methane is CH4, very simple organic compound. But they live in swamps. So they're important for herbivore digestion, too, because they can actually live in the gut of cows and help them digest methane. So methane is the production of the byproduct of digestive materials, and these methanogens can actually metabolize that methane and use some of it for themselves, and, right there for the cows. That's a symbiotic relationship. They can live really salty. That's that picture in the lower right. When they live in these really different salty and environment conditions, different species grow more or less closer to the extreme environment. They're different colors. So sometimes there'll be bright greens and yellows and oranges, and those are all just different species of bacteria with different species of archaeans or prokaryotes that can deal with different conditions, extreme conditions. Some of them can live in those really extreme conditions. That's really not a good picture. That's a cold smoker, so that's probably more, that's not hot water. The optimum temperature for those that live in those black smokers, I showed you the picture earlier, and there's the gray smokers, about 60 to 80 degrees Celsius. Remember, 100 degrees is boiling, so they can live down there in these places. Of course, the problem with the cool smoker would not be the temperature, but as much as the noxic gases and hydrogen sulfide that comes up. Remember, like I said, there are sulfur bacteria that can live on and stuff, so plenty of them. This last group are really cool. The nanoarchaeota and the Prokaryotes, they're really cool for another reason than the Igniococcus is one of these nanoarchaeota. I lumped them together, okay? There's two weird groups, but one of them, and I believe it's a nanoarchaeota, is an obligate symbiote of another archaean, Igniococcus. So on the right-hand side is Igniococcus. There's the host, and the little brown ones are the nanoarchaea. So we got archaeans associated with another archaean, and that's kind of cool because, I mean, we even down to that very basic level of extremophiles, we have this idea of these symbionts living together. I don't remember if the relationship is parasitic or if it's a commensalistic relationship, but it's a symbiotic, it's an obligate, and they have to have it. But really the cool part of all this is there's only one species on Earth found in only two places. One single Atlantic hydrothermal vent in this pool in Yellowstone. The Prokaryotes, the other ones, which I don't have a picture here, they're only found in Obsidian Pool. That's the only place they're found on Earth. That's a word we call endemic, right? They're only found in those places. That's really pretty cool. So that very extreme environments, very bizarre organisms that live, they're very rare. Now, are these some of the remnants of some of the organisms that help populate? No, not necessarily, probably not. But they do show us that there are critters alive even today that can deal with the environment that was very common back then. And very common on other planets, by the way. Right? So, never know. The good and the bad, let's do the bad first. Those are the pathogens. In the early 20th century, bacterial infections killed about a fifth of children. We didn't know they were coming from, right? Because we didn't have microscopes, especially kids younger than 5. One of the big ones was tuberculosis. There's a picture of it down below. It affects the respiratory system, and for thousands of years, it was a scourge, wasn't it? Wiped out huge, massive amounts of people. It's one that's spread from human to human, and it's very easily spread in concentrated conditions. Well, we came up with many different types of antibiotics throughout the years to treat not only tuberculosis, but other bacteria. But if you know something about their evolution, which you do now, you know that sometimes what you can do is actually encourage adaptation to that antibiotic. So if you keep using the same antibiotic, you probably don't kill them all, right? There's a lot of them. So maybe 1 or 2% survive. They reproduce, and maybe they survive because they got a plasmid, right? A plasmid that makes them immune to your antibiotic. And then the other ones that don't, maybe they start doing that sex pili swapping and the conjugation thing, and before you know it, you've got a bunch that are immune to your antibiotics. What do you guys switch your antibiotic? Doctors are a little less inclined to prescribe antibiotics now for common cold, letting the immune system take over in the hopes of kind of limiting that type of artificial selection is exactly what that is that's going on among the healthcare, right? This is salmonella, causes typhoid fever. Today, between 16 and 33 million cases of this are re-emerging. So another one that's coming back probably because of resistance to antibiotics. 200,000 deaths a year. The cares can be asymptomatic. So here we got a lot of problems. We got a re-emerging disease, which explains that it's, which could be because of some artificial selected resistance to it. Salmonella is a really horrible, gut disease to get, a really horrible digestive disease. It's re-emerging, and the cares are asymptomatic, so it's a silent disease, right? Of course, when we think about bacterial diseases, some of the kings are these guys, the Black Death. There it is, Yersinia pestis. The 6th to the 8th century, maybe longer. Another breakout in the 14th century, one in the mid-17th century in London. There's some pictures in the plague of London. Just pick up the vibe. This is where Monty Python gets a lot of it. Bring out your dead. Those are pictures up above that were drawn back in that area, just kind of illustrating this thing called the Black Death. It was a pandemic. Pandemic means it spread all over the world. The vectors were a prokaryote that was living in the rodents. People got tired of living around rodents, so they got involved in these big projects in these cities. The cities were getting bigger back then to get rid of the rodents. The fleas said, well, now I got nothing else to eat, so I'm going to jump on the humans, right? You know the story about the Pied Piper, right? He was paid money to play his pipes and lead the rodents out of the city. And then I think maybe he got screwed over on the payment or something like that. But the rodents being let out of the city. A lot of our poems ring around the rosies, pocket full of posies. People were dying back then so much that people carried flowers, posies, in their pockets in order to deal with the smell, the stench, because the cart hadn't come around and picked them up. Bubonic plague arises up every once in a while lately, but we pretty much got a handle on it with antibiotics, so hopefully no more pandemics, but there are plenty of other bacteria, especially ones that we may be artificially selecting. They're on the verge of one of these. Horizontal gene transfer, that's some of the biggest problem. Virulent strain is a death-causing strain or disease-causing strain. Non-pathogenic. So virulent, non-virulent, pathogenic, non-pathogenic. So they'd be synonyms, right? Griffith's work that you talked about, remember the dead R picked up the genetics. It's one of the ways that these bacteria can get genetic information, right? Through transformation, through absorbing it from the environment. So it picked it up and it developed the ability to become virulent. And in the case of the stuff that Griffith was working with, the one that was virulent had a capsule. So the capsule aided it to hide, enabled it to hide from the immune system of the mouse. E. coli. Now, we all have E. coli. E. coli is a normal microbioflora of the lower intestinal gut of warm-blooded vertebrates. The problem are the strains of E. coli, okay? Or they call them serotypes. Serotypes are kind of like breeds. So, and I think I have this right, the O157 strain may have passed to the H7 strain or the other way around. But anyway, those two strains got together And the fimbriae are not found in the non-pathogenic strains. Those two strains of E. coli got together, and the non-virulent strain picked up the ability to be virulent, to have those fimbriae through what we believe is probably conjugation, right? Passing up that information. Or it could have been through transduction. It could have been a virus that did it too. But that's where the problem is we have with lettuce. The problem, the way E. coli gets on lettuce is because E. coli is a normal resident of lower intestinal gut of warm-blooded vertebrates, cows poop. In their poop are going to be E. coli, many of them good ones, but if for some reason one of these strains has become virulent, then the feces washes into the waterway, and a little further downstream, maybe Farmer Brown waters his lettuce or spinach with that water. And normally it's not a big deal because the E. coli are harmless, right? But there's some virulent strain going around, so that's why they try to trace it back to where it is. They say we get food poisoning more often than we really think about it in a year, but there are good bacteria. There are the ones that are the nitrogen fixers that we talked about. The biological nitrogen fixers, only ones that do it are the prokaryotes, which is obviously important, right? It's the second most important or biological process on Earth after photosynthesis. That's amazing. Without the bacteria, you wouldn't be able to do any of this. You wouldn't have plants. The intestinal microflora, and I got a little picture on the right. You can see the numbers, tons of different species spread throughout your gut. These are all important bacteria. They aid you in gut and digestion and that sort of stuff. Many times bacteria perform a good function against other bacteria because they just take up space. So if you got these good bacteria living there, and maybe they're commensals, right? Maybe they've got a benefit living around in your gut. They don't bother you. They don't help you. They don't bother you. But what they do, this is kind of stretching the commensalistic part a little bit, but what they do is they take up space so bad bacteria can't find a place to live. So sometimes it's simple as just finding a place to live. Bacteria have such simple genomes, we can trick them into genetic engineering. We can stick genes into them and have them make stuff that they don't need, like polyhydroxyl alkylamate. It's a biodegradable plastic. That's what you see in the picture. Those little lumps are PHA. The bacteria doesn't need it. Well, it's kind of like a virus, and sometimes that's what we use as a virus. We put the information we want, how to make this stuff, into a virus, and then viruses go through transduction and inject it into bacteria. Then we grow up a bunch of bacteria. It's how insulin's made too. We inject the bacteria we inject the bacteria with the insulin gene. Bacteria don't need insulin, but they make bunches of it and spit it out, and then we use it, of course, for folks who have diabetes. Bioremediation is using life to fix our mistakes. That's what this guy's doing. This is the Exxon Valdez spill. Of course, we're more familiar lately with the one that happened back in the summer of 2010. But what they do, he's spraying. In some cases, they just use physical hot water, spray it off the rocks, which doesn't do much. But in some cases, they spray a mixture that's got bacteria living in there. There are bacteria that actually live on oil. They feed on oil, remember, so they can eat anything. And if you're thinking to yourself, where did they get the oil before humans were around? Oil, a lot of the oil in the ocean, a good portion of it is naturally leaking into the ocean. It's not our mistakes. It's just there. So they're critters who would take advantage of such a rich hydrocarbon as oil, right? So we have taken those and genetically engineered them, and then we put them in these solutions and spray them out and hopefully digest the oil. The problem with the Exxon Valdez, from what I was reading, is even that happened 25 years ago. Even if you dig down a certain depth into that area up in Prince William Sound, there's still oil. It takes a long time for oil to go away. So the oil in the Gulf oil spill, it's still there. That dispersant that they were using, part of what a dispersant does is it takes big drops of oil and make them smaller, so you just don't see them. And then there's some oceanographers. Holy cow, it's early in the morning. That believe that maybe some of that oil has descended down into the water column. So instead of it floating at the top, because of the way it's down, so there might be these huge pools of oil that organisms have to live with at depth. So it doesn't go away real fast, okay? So it's one thing we have to keep in mind as we continue our use of fossil fuels, which we hopefully won't forever. So, all right, that's my chapter for bacteria. If you have any questions, give me a holler.